Good morning, everyone. I am Dr. Manisha Nanda. On behalf of Department of Biotechnology, Dolphin PG Institute of Biomedical and Natural Sciences, I welcome you all to the webinar on plant molecules for human health and plant defense signaling. First of all, I express my gratitude and welcome Dr. Mihir Kumar Mandal, our guest speaker for today. Thank you, sir, for sparing your valuable time to us. And now, I take the privilege of introducing our guest speaker to the audience. Dr. Mihir Kumar Mandal is working as Assistant Project Scientist in the Department of Plant Pathology, UC Davis. Dr. Mandal was awarded PhD in Plant Pathology by University of Kentucky in the year 2012. Dr. Mandal has also worked as an assistant professor in the Department of Biology, Claflin University. He was also a research scientist at University of Georgia and US Vegetable Laboratory. He is an active researcher in the fields of plant microbiome interactions in vegetable crops, molecular genetics, genomics, metabolomics, development of pesticides. He has published his work in various peer-reviewed journals of international repute. Dr. Mandal has been recognized for his research contributions by the Government of India as well as various international organizations. So, without any further delay, I invite Dr. Mandal for his talk. Sir, please. Over to you, sir.
Thank you, Manisha. A very nice introduction for me, and I would like to thank you and Dr. Sanali Singh for the invitation and giving me this opportunity to present our research. As you see, the topic is like plant molecules for human health and plant defense signaling. So generally, when we talk about different plant molecules, we consider different phytohormones and other small molecules which are associated with both defense signaling and also how their intakes can improve our health. So before going into detail of my project, I would just like to give a gist of my previous experience and my education, just to have an idea like how I came from my background from agriculture field from India to US. Like I started I come from Dhanbad, where is the Indian School of Mines, and then I went through the ICR program to do my undergrad at Assam Agriculture University, Jorhat, and then I went for my master's in biotechnology program at GB Pant University and Agriculture and Technology at Uttaranchal, where I met Dr. Singh. So that's how we know from a long time. Well, I think more than 20 years. So thank you, Dr. Singh, for the invitation, and very nice to interact with you during this last 2 weeks. And from there, I moved to JNU, and there is a National Center for Plant Genome Research, and I started my career as a project scientist come PhD program it was. So I stayed there for a couple of years, and then I moved to University of Kentucky, Lexington at USA. So I did my PhD on lipid signaling, how lipid signaling can help in plant defense properties. So like here you can see like it's a wild type Arabidopsis plant, and then you have a mutant. So it's a EMS mutant line, and this is smaller in phenotype. And this smaller phenotype have constitutive plant defense, means this plant is resistant to so many pathogens because it have a molecule which is, or a phytohormone, which is very high in level. And that is like salicylic acid. So you might have heard about the term salicylic acid. So from there, I moved to Virginia Tech, a very nice university and lot of nice buildings. And so I worked there as a postdoctoral associate in the genomics lab. in both Department of Biochemistry and it's a forest genomics. So I work on different pathogens like Phytophthora Fruit Rot and chestnut blight. And from there, I moved to South Carolina, which is like the eastern part of US, and it is more close to the Atlantic Ocean. And there I worked at US Vegetable Laboratory. It's a USDA, United States Department of Agriculture, and mostly my research was focused on molecular breeding, genomics, and metabolomics in vegetable crops. So I worked most on the cucurbitaceae family crops, mostly like watermelon, zucchini, or squash, or cucumber, all those crops. So mostly I try to find out any gene which could be responsible for resistance to those crops, or finding any metabolites which could be helpful in providing those type of resistance. And then from there, I went for my assistant professor position at Claflin University. So I had my own lab, and I still continue with some of my research on those parts, like mostly on developing biofungicides against some of the fungi. And from there, I moved to now in the West Coast last year, like in August. So me and my whole family, like my wife also got a job here in a company. It's a saccharotic acid company. It's a big company here. So we all moved here. And so we are now located at California. It's in Salinas location. So most of this, it's just close to the Silicon Valley. So we are just close to the Silicon Valley. And then here I work on mostly on lattice verticillium and the soil microbiome. So here we are more dealing with how the soil microbes can help in better growth of plants. Like, you know, in the soil, you may have some beneficial microbes, some maybe pathogenic. So we try to identify what are the beneficial microbes which can suppress the pathogenic microbes, and whether those can be helpful in the long sustainable crop production. So overall, my research is like directed towards plant pathogen interaction, where we talk about resistance protein, which are associated with plant, and then the AVR effector, or the virulent protein, which are more associated with the pathogens. And then also I worked on some part on the nutritional genomics which deals with different fatty acids or the lipids, and also the antioxidant molecules. And for this, I use different technology like molecular biology, breeding, metabolomics, or genomics or CRISPR-Cas9 type of technology, which helps in improving the host resistance and also like improving the nutritional status of the crops. So before getting into the management of the crop, I wanted to give an idea what is host resistance signaling. Some of you may be aware of it, and some of you maybe it's a new topic. So what happens, like whenever a pathogen attacks a crop, the pathogen have the AVR effectors. So they secrete these effectors or the proteins, which could be recognized by the R, which is like a resistance protein, and which is present in the plant or in the crop. So this AVR can be recognized directly or indirectly by the crop resistance protein. And then you have a signal, you know, like we sometimes get infected by a bacteria or a virus. So our body have the capacity to recognize those antigens through our antibodies. So same thing happening here in the plants. So then you generate a signal. So that first signal is the HR symptoms, which we call as hypersensitive response. And this can give rise to a local defense response to the leaf or to the plant. So if a plant or a leaf is infected by a pathogen, you see a defense in that leaf. But it's not limited to this localized defense. This signal also moves to the systemic leaf or the nearby plant, or to any of the vegetative or reproductive part of the plant. So that movement is called a systemic acquired resistance. So what happens if this signal moves from here to the another neighboring leaves? So if a pathogen is attacking this leaf, maybe the primary pathogen this, or it could be a secondary pathogen. But because of the signal is already available, so this leaf is already tolerant or resistant to the primary or the secondary pathogen. And this is known as SAR, systemic acquired resistance. So as we say, there could be a direct or indirect interaction between AVR and R. So what happens if suppose this interaction happens? So if this interaction happens between the AVR and R, then you see the symptoms in the upper panel. You see the plants are resistant to different pathogens. So I work on all these pathogens here. So these are powdery mildew pathogens. So you can see in the susceptible plant, you can see the powdery growth of the pathogen. Downy mildew, this is another type of pathogen, oomycete pathogen, and you can see it have a different type of symptoms. These are the cucumber leaves, and you can see this yellowing of leaves. And then you have the Phytophthora Fruit Rot. This is a watermelon fruit, and you can see this fruit is resistant, but however, this fruit is susceptible. So this is like a very, again, this is a very devastating fungus in US and also in other parts of the world. So what growers do, like they have to more dependent on the different type of fungicides. So they have to spray the whole field sometimes, and it costs a lot for the growers. And generally, we need to find out something which is more sustainable and which is more environmental, eco-environmental. So it's like more not toxic to the environment, not to the soil, and not to the human health. So what we do here, we try to find some sustainable approaches. So one of the approach that we look for here is like, I showed you in the previous slides, this is like the powdery mildew disease. So some plants could be like tolerant, like this is called as innate resistance. So the plant itself have resistance, whereas the other one, which is susceptible. So what we try to develop here, we try to use this tolerant character and incorporate into this susceptible plant. How we can do that? So one of the way we do is like grafting. So you may have seen a rose plant may have different color of flowers. So we use the same technology here. So what we are doing here, we are doing the grafting approach. So you might know what is grafting. Like it's like a joining of plant parts together, and maybe from different varieties or different color of varieties with different color of flowers. So you can join the branches, and you can allow them to grow together. So maybe in the next year, you may have multiple flowers in the same plant. So same thing here we do here. So the upper part of the plant we call a cyan. So this is a susceptible variety. And this is the resistant variety, which is like more, it's bound with the soil. So that is called as rootstock. So this is rootstock, and this is the resistant line. And the susceptible one, you join with the resistant one, and then you put a clip, or sometimes you use paraffin and just let it grow. And slowly the cells join, the tissue joins, and they can grow together. So what happens when you join both the rootstock and cyan? So this is an experiment what we did. So this is the in the right panel, what you see, like this, you have the resistant rootstock, and a susceptible host is grafted here. This is the susceptible rootstock, and again, a susceptible cyan is grafted here. And when you spray the pathogen into both plants, what you are observing here, the plants which is grafted with the susceptible rootstock is susceptible to the pathogen. But when you grafted with the resistant rootstock, you see a resistance phenotype in the susceptible host. So how does that resistance comes into the susceptible host? So what that indicates that the rootstock is providing something as a sink, and it helps something is moving from this rootstock to the cyan, and that's providing a defense mechanism to the susceptible host. So this plant should have look like similar, but we don't see when it's grafted on the rootstock. So you can see a contrasting phenotype in the both susceptible cyan when it's grafted with the resistant rootstock and with the susceptible rootstock. So we wanted to identify what's providing that resistance. So what molecules are involved in that type of resistance. So one of the way is like you do genomics or you do metabolomics. So metabolomics is like more on like identifying different type of metabolites, whereas genomics like you do sequencing of the genome, but here the genomes are same. So both science are same genome. So it's the best way to identify the molecule is to go for a metabolomics approach. So what we did, so we collected samples. We collected samples from this part of the hypocotyl and also after the joining parts. Again, here also, we collected samples from here and also from here. So if we go to the previous slide, so this is like we collected samples here and also from here after the joining parts. And we wanted to see what molecules are moving in that joining grafted rootstock and sign. So the PCA plot, let's go to the upper panel. So what we see here, this is a resistant, So this is the rootstock profiling in the upper panel. So we also did like just the host plant, so the resistant plant and the susceptible plant. So in both cases, what you see, like different metabolites are expressed. And also in this is the like metabolic profiling of the cyan. So this is like Mikili, one of the susceptible host, and which is grafted on the resistant line PMR. And again, the Mikili is grafted on the susceptible rootstock, which is the powdery mildew susceptible Citrullus lanatus line. So you can see a different type of metabolite profile between the two susceptible hosts, either grafted on rootstock, resistant rootstock, or susceptible rootstock. So you see that there is differential regulation of those metabolites when it's grafted on the resistant rootstock and when it's grafted into the susceptible rootstock. So altogether, we identified like 27 metabolites. So we didn't find any qualitative differences. So we identified that all 27 are similar, but we see a quantitative differences, like the amount of those metabolites. And just I wanted to give a brief how does metabolomics work. So you collected the samples, like we collected the samples from the hypocotyl, from the rootstock, and also from the cyan region. Then we run through the NMR instrument, we collect the data, we do an analysis of the data, we do the metabolite identification through different software, which is associated with those instruments, and then we try to identify some marker which are very differential. So this is our sample. We collect the hypocotyl, and then we quench the metabolomes with liquid nitrogen. So when you collect samples, we freeze the samples in liquid nitrogen. Anything, either you extract DNA, or you extract metabolite, you make sure the metabolite doesn't, like, even an wounding can change the metabolite profile of your sample. So you make sure when you collect your sample, just dip into the liquid nitrogen so that there is no change in profile after your collection, and that it remains similar to what was available in the plant, intact plant. And then you do the lyophilization to remove excess of water from those samples. And then you add a solvent of methanol, chloroform, and water. So this is a mixture of to identify both polar and non-polar molecules. So you centrifuge it, and then you separate the polar and the non-polar. So the polar comes in the top surface, whereas the non-polar comes in the bottom, and then you have the protein precipitates, which are in between the polar and non-polar. So the polar, you can directly use the NMR buffer and put into the NMR instrument. And the non-polar, you dry the non-polar and then again use the NMR buffer. So in this way, we identify both polar and non-polar products. So this is the NMR instrument that we have used to identify all those molecules. So this is a representation of the 1D NMR spectrum. You can also do 2D NMR spectrum. Sometimes, like when you, it's very difficult, like you see most of the sugar molecules peaks are very high. So you may have an overlapping peaks. So in that case, we do go for the 2D NMR to find out those metabolites if there is something like overlap. And most of the you see the most of the amino acid profile which are like separated quite easily, but when you have a glucose, you need to can go for different steps of purification, or you can go for 2D NMR. So what as you remember, we identified 27 molecules in that process. So out of that, 6 of them were quite differentially regulated, out of which the melatonin, one of the molecule which was like highly upregulated. So generally, what is melatonin? Like, it's a pineal gland hormone, and many of you know that people sometimes the doctor recommends to use it if you have a sleeping disorder, or like if you have a jet lag while travel, or sometime if you have an insomnia. So the doctor usually prefers to have this melatonin. So we couldn't understand like how does a melatonin which is produced in the plant could provide that type of resistance. So one of the hypotheses is like to check, is this melatonin is really giving the defense. So what we did, like we bought the melatonin through Sigma company, and then we spread the melatonin on the different crops. These are the different leaves, like this is from cucumber, this is from squash, this is from watermelon susceptible line, the PMS, and this is a resistant line, PMR, which we already discussed in the previous slides. And this is the control, which were initially sprayed with water. So you have two types of treatment. One is sprayed with water and one is sprayed with melatonin. And after a couple of days, you spray the pathogen on both crops. So you can see a distinct phenotype, like in the control, which were sprayed with water. You can see the growth of that pathogen, this powdery mildew growth on most of the plant, except the resistant, because the resistant is itself have an innate immunity. So that's why you don't see any growth of the pathogen. So this shows and prove our hypothesis that yes, melatonin is indeed an important component for resistance against powdery mildew pathogen. And the same thing, we also we also wanted to see that if this have a broad spectrum resistance, is it limited to powdery mildew, or can we provide protection to different type of pathogens? And also, this pathway is quite conserved in plants, and so we also identified the whole pathway in watermelon. Like, this was mostly related to watermelon, so we identify all the genes and also localize the genes associated with the production of melatonin. So we especially look for the SNAT, which is one of the regulator of this melatonin biosynthesis, and also the SMT gene. So one of them is located in chloroplast, and one of them is we found that it's present in cytoplasm and also in the nucleus. So these are like you can what we do, like you can fuse the gene with the GFP tag and express into an agriculture, and then infiltrate tobacco leaves. So these are the infiltrated tobacco leaves with the GFP tag or the red flag RFP tag, which is a red tag protein. So that's why you see here the chloroplast is localized to the chloroplast, and this is localized to the nucleus and cytoplasm. So this means most of the melatonin biosynthesis is happening in the chloroplast, and also its regulation happens through the nuclear regulation and also the cytoplasm movement. And the structure is quite conserved. We find the genes are quite conserved, but different type of varieties have different isoforms. And you can see like some of them have a big N-terminal domain, and some of them can have a truncated domains. It's quite conserved among different plant species. So we looked through all the different plant species and did a phylogenetic analysis, and it's quite conserved across different species. So this is like when we spread the plants, we see a resistance. So what happens if you transform that gene into the crop? So generally we don't prefer much of transgenics nowadays after coming the CRISPR technology, but before that we were working on to identify if really that happening endogenously in the plant. So what we did, like we transformed the gene, the 35S SNAT gene with the GFP tag vector, and then we transferred into the watermelon. So we did the transformation technology into the watermelon, and this is the way how we developed the callus, and then we transformed it through agrovector medium transformation. And then we looked through all the transgenics, like we got like 4 transgenic lines, and these are the 4 transgenic lines, and this is the transcript, changing transcript after transformation. And we see a reduction in the growth of the pathogen as compared to the susceptible, but we didn't get a complete resistance. So that could be because there could be another factor which could be involved in resistance, complete resistance, but this gave us a partial resistance as compared to the susceptible host. So you can easily see that we counted the fungal colonies, so pathogen colony, and you can see the resistance is already resistant, whereas susceptible have high conidial count, whereas the 4 transgenic lines have a reduced conidial count. So how the melatonin mediated defense signaling works in plant? So what's happening in the plant? What's the networking, or how the signaling goes from upstream component to the downstream component? So one of the way of doing that is doing RNA-seq or transcriptomic analysis to find out all the differentially expressed genes, or we call as DGs, under melatonin treatment. So what we did, like we have the susceptible host, and then we sprayed melatonin, and we also have the resistant host. So we did RNA-seq analysis, and we identified there are 1,388 genes which are differentially upregulated, out of which like 796 were upregulated. So we try to identify what are the genes which are becoming upregulated, and we found that some of the R genes, we remembered from the pathway, like when an R or AVR interacts, then the signaling happens and the hypersensitive response. So we found that some of the genes were quite upregulated, and 10 of them were quite upregulated both under innate conditions, like in the resistant line, and also when you spray the melatonin. So we are still working on the CRISPR to identify like if we can mutate those genes and see if that provides a resistance or susceptibility. So overall, the melatonin pathway model works like this way: like you have the fungi or the omycete pathogen, and this pathogen produces a hosterium, and that releases the effectors. And this effector can be either secreted into the cytoplasm of the plant host, or sometimes the fungus itself can secretes those pathogen-associated molecular patterns, and these are like the secretory proteins. And these secretory proteins can be recognized by the R proteins. The R proteins are not only available in the cytoplasm. So like here you can see one R. You can have a different type of R which is present in the cell surface. So those PAMPs can be directly recognized by the host R proteins, or sometimes it can be recognized through the cytoplasmic R proteins. So you can have a like effector trigger immunity, or you can have a PTI type of immunity, which is based on the presence of PAMPs. And this type of both regulation could lead to a increase in the c-KIT pathway, and this c-KIT pathway leads to an increase in melatonin. So you saw that there was an increase in melatonin production when the pathogen infect the host crop. And this melatonin can increase the assay level, like the salicylic acid. And this salicylic acid overall governs the resistance through nuclear regulation of different work keys and transcription factors, which we found through the transcriptomic analysis, and which ultimately gives the direct defense to the host crop. So now we know how melatonin works in plant. So now we go into the human health pathway, like how it helping to us, like it's a pineal gland hormone, and why we need it. Like we need for like if we have a sleeping disorder. So can we always directly take pills, and can that gives an immunity to our body, or there's another way or sustainable production of crops that can help you giving a different type of immunity? So melatonin is an antioxidant molecule. So if you My work in mostly at USDA, US Department of Agriculture. We know that this can provide resistance to grafted plants based on our previous slides. Also, it have high antioxidant levels. So the watermelon that we eat have high lycopene and beta carotene. Both are antioxidant molecules. And it also gives resistance to the Phytophthora based on our study. So we don't know what's happening in crops or it's happening in watermelon fruit. So how can we know how much level of melatonin is present in plants? So it's the same way you have to do the metabolomics approach, and you can use the either NMR or sometimes you can use the LCMS or the mass spectroscopy, and you can find out how much melatonin is present. So my work in US Department of Agriculture was mostly focused on the finding out the different antioxidant levels. And watermelon, we all know it's a rich source of vitamins, amino acids, especially the sertraline levels is quite high, and it have different type of ions like potassium, magnesium, copper ions, and rich in lycopene and beta carotene. So I tried to identify the level of melatonin. So we used the same NMR technology, and we did the metabolite analysis, and you can see the PCA plots. We did like almost tested 30 different varieties of watermelon, and you can see the metabolite profile of each of the 30 varieties with 6 replicates. So all replicates are showing here in similar color. So you can see that metabolites variations in some of the profiles. So overall, we found there are 5 varieties. I think I removed the slide. Okay, there are 5 varieties which are like similar profile, and we find that independent of the sugar content, like different watermelons have different sugar content, or we call as Brix content. So irrespective of the Brix content, we found 5 watermelon varieties which have similar profiles and could have lead to a resistance to plants. So 5 varieties have resistance to different pathogen, and also we identified the levels, and we found that this level is quite variable. So you can see a variable of like 48 nanogram to as high as 760 nanogram between different varieties. So if you know which variety have high level of melatonin, so you can eat that variety instead of using a medicine. So we know now which variety have, and sometimes you can also use for breeding purpose. So if your variety doesn't have a high level of melatonin, you can use the high melatonin content watermelon line to breed with a low melatonin content watermelon line. So in order to further prove how this melatonin could be helpful, so what we did, this is in one of my collaboration with my Claflin colleague. So what we did, we tried to find for an undergraduate student, we did this research for studying the effect of melatonin treatment on cervical cancer cell lines. So we used the HeLa and C-HeLa cell lines, and we treat them with different concentration of melatonin from 1 micromolar to 100 micromolar. And we had the control with 1% DMSO because the melatonin is not soluble in water directly. So use DMSO to make sure it's not the effect of DMSO, it's just the effect of melatonin. You use a separate control to check those treatments and your overall results. So we use 2 types of cells, the HeLa cells, which have the HPV 18 or the HPV 16, the C-HeLa cells. And you can see the picture, not so clear visually here, but you can see A is the untreated C-HeLa cells, and this is the HeLa cells. So you can see D, which is the HeLa cells, and the C-HeLa cells, which have a treatment of 100 micromolar. You can see a structure of the cells are coming back. So you can see the phenotype of the cells in a better shape. So this shows that with the help of melatonin treatment, you can reduce the cancerous activity of the cells. So the cells are coming into a better shape, into a normal shape, as compared to the cancerous cells. So in order to further prove what's happening in the normal cells due to melatonin treatment, we checked the different expression of the tumor suppressor genes. So we checked 3 of the tumor suppressor genes, the TP53, TP73, and RASF1A cells type of transcription. So what we found, you can see with an increase in 100 micromolar melatonin, you see an increase in the tumor suppressor cells TP53 gene, and also the TP73 as compared to the normal HeLa cells. And also in the case of RAS, the increase was not that significant. So we don't know why this, we did just one time. So we are not sure now why it didn't increase, but we know that 2 of the tumor suppressor genes or the transcript were induced in case of 100 micromolar concentration. So this gives an idea that with the improved melatonin level in your body, either through you take a medicine or you take through fruits or vegetables which have a high melatonin level, could reduce, can provide more antioxidant capacity to your cells and can reduce this type of cancerous activity. So in overall, we can say that melatonin is a mobile signaling. Why we call it as mobile? Because you remember we grafted between a scion and a rootstock. So from the rootstock, this molecule moves to the susceptible scion. So that's why we call it mobile. It can move from one part of the plant to another part of the plant. So, and overall, it can provide a plant defense, because you saw when we spray into the leaves, it can provide resistance to powdery mildew. I didn't show you one pictures where if you spray the melatonin, and you can, in fact, with the Phytophthora, it can still provide resistance to the fruits. Melatonin provides broad spectrum resistance against different oomycetes, so it can provide resistance to Phytophthora. It can provide resistance to powdery mildew. We also identify 5 watermelon varieties which have identical metabolites, and they can provide have an impact on disease resistance of those metabolites. Melatonin levels are quite variable in different watermelon lines. So you saw that in those 30 different varieties, there was change in level of watermelon. So maybe the watermelon that you are eating could have a high melatonin. So it's good to select a variety which could have high melatonin as compared to a normal level of melatonin. Again, finally, the melatonin treatments at 100 micromolar concentration may help in changing the morphology of the cervical cancer cell lines, like the HeLa and CHeLa cells. You saw that picture. So, so it could be like melatonin have an effect on bringing back the cancerous cells into normal cells. I also worked on, this is like one of my PhD work on, I worked on, I just wanted to show you what I have done in my research, some of the extra work. So just in one slide, like you can localize, if you want to know a protein, or if you're studying any genes or plant genes, you can always localize them into a cell. So you see the plant cells, which are the GFP fluorescent plant cell, and the red one is the nucleus. So this is the plant cell with a nucleus. So I tried to infuse different genes and with a GFP tag or a RFP tag, and you can study the function of different genes in a plant. So this is like one of the nitric oxide synthesis gene, and the SI2, which you remember the small phenotype plant because of a mutation in the sterile desaturase gene. So, so you can see that both nitric oxide synthesis gene and SI2 gene can co-localize at the same place of the chloroplast. Also, one of the work I do here, like sometimes you don't want to use fungicide to control a pathogen. So what is the other way you can control pathogens? Sustainable approach, look for soil microbiome. In your soil, there you can find some bacteria which could be beneficial and can suppress your pathogen. So this is like a bacteria we isolated, and where we can see that this is the pathogen, the Phytophthora This is the normal plate, how it grows. If you put the same agar plate and you streak them with 3 corners with a bacteria, you see there is no growth of the pathogen. So it inhibits the growth of the Phytophthora here in this plate. These are the other different type of isolates, and 2 different type, and we identify more than 10 isolates here. So this shows that you don't have to spray fungicides to have a crop or to have a food, which have like both an impact on your human health and also impact on the environment, also impact on the soil. So you can identify some bacteria, and you can find those metabolites, and you can spray in the field, which are more sustainable instead of fungicides. So this is one of the project I got funded in the 2020-21 year. Also, since I was busy with some teaching and research, so I always prefer a student to bring into real field and take a look, like bringing to have an expertise or bring some expertise in the field who are growing crops, and they can let you know how they do different type of crop production, what they use, or sometimes you can bring them to the lab and they can show you the different instrument and expose them. This is like undergraduate students, and it's good to have that type of exposure. Like you can take your student to industry if you have a close by industry, or you can have a close by farms. You can always have a given hands on what's happening in day-to-day nature. I use different type of tools for my teaching. Now I'm like totally on research, now back to research, but when I was in Claflin, I have both teaching and research. So I use the different models like Jove or Camtasia or Moodle. Mostly I use the Moodle, Jove, and the Labster virtual lab. So like if some, due to the COVID situation, it was most of our classes were virtual. So Labster virtual, like, the most of the lab technology, like running a PCR machine, or running a NMR or so running a jail, PCR jail, you have all the information through those virtual tour. And this is like, I work on one of the NSF funded project. It's a National Science Foundation. So what we do, like, the beans, these are the different type of beans. So, and you can find the beetles, in those beans sometimes. So what I try to find, like, if you crush those beans, the beetles, you can find different type of bacteria or different type of microbes inside the gut of that beetle. So even identify biocontrol from an insect gut, and that can also protect the crops from those pathogens. So this is another way of sustainable approach. You can identify some biocontrols from an insect also to produce as a biocontrol or biopesticide. Overall, I just want to acknowledge to all my funding agencies, my different collaborators, and I'm very grateful for Sekhar, who is in USDA, US Department of Agriculture, Charleston. So most of the work what I showed here is in his collaboration, and I also worked as a postdoc there also. So most of my projects, what I've shown here, is through Dr. Sekhar Kaushik's lab. And currently I'm working on lettuce, which is mostly it's a leafy vegetable. So we try to identify some soil microbiome, and also like how to control the verticillium disease in lettuce. Like in California, it's a big problem here. My special thanks to Dr. Salini Singh and the seminar committee and everyone for the IT people who has helped me in this presentation. Thanks. And since I work on watermelon, my kids love watermelon, so it's good to present. But now they have grown up, and I would be happy to take any questions.
Thank you so much, Dr. Mihir. It was so nice to hear your research work. Any questions? Anyone? I don't think they have posted. Any piece of advice you would have in which you would like to give to our students, Dr. Mihir?
Oh,
These are
sure.
the MSc students, please.
Yeah, I removed some of the slides. Sorry, it's such a big file, so I have to concise in 50 MB, and I made PDF instead of PowerPoint. So anyone, if anyone interested in doing PhD here, like if in UC Davis, always welcome to contact us, like me or Dr. Subarao, where I'm working here. So, and I can send you the link and everything. So there's I think it's still like in between, due to COVID, there was some university didn't allow GRE at that time, but I think again the GRE may have to do. Some of the students may have to take the GRE, and like I always prefer, like if you are interested in going into companies, there are a lot of companies coming up. I always look for advertisements, and there are different websites which like have all the informations. So like here we have seed.com, and that gives most of the seed company informations if there's any vacancies here. And if anybody wanted to go for postdoc, can contact me. I know few of the people who looks for postdoc for good students, so always welcome.
Okay, thank you so much for your kind words. I, Dr. Shalini Singh, on the behalf of Biotechnology Department, extend a very hearty thanks to Dr. Mihir Mandal, who honored us with his presence and took out his precious time, valuable time for this webinar. I
Thank
especially like to mention your efforts, sir, for briefing about your research work so precisely to our students. I also wish to express my gratitude to our principal ma'am, Dr. Shalja Pant, and our chairman, Sir Shri Arvind Gupta ji, who has always providing us encouragement and support for such events. I heartily congratulate to all the participants who have attended this webinar, and surely they will take some experience from it and will enrich themselves. And no program can be successful with a single person. Along with Dr. Meher, I extend my big thanks to our technical team and my fellow colleagues, Dr. Manisha Nanda and Ms. Dipti Gulati, for making this event successful. Have a nice day, everyone. And Meher sir, good night to you. And thank you for sparing your time. Thank you so
Yeah,
much.
thank you, Dr. Singh. Have a nice day. Bye
Okay.
bye. Take care.
Thank you so much. Bye bye for now.